Today we're going to look at Chapter 16 or Lecture 16. It will correspond to the Bauman text, Chapter 16, or the tutorial text, Chapter 17. Here we're looking at adaptive immunity and specific immunity. This is the body's ability to recognize and defend itself against specific invaders, their products, as well. This is a dual system. It's going to involve both humoral and cellular components. So there's 5 attributes that are associated with adaptive immunology. First is specificity. Reactions are going to be precisely tailored to an attacker. Inducibility, which means specific antigens are going to induce cells of adaptive immunity. Clonality, these induced cells will proliferate for many generations of clones. Unresponsiveness to self, it does not act or target normal body cells. And then memory, it's going to adapt to respond faster and more effectively in subsequent encounters in the future. So we'll look at the elements of adaptive immunity, your tissues and your organs. First is the lymphatic system. The lymph is going to be a colorless watery fluid that arises from the interstitial fluid. Your lymphatic vessels are going to be a one-way system that are going to conduct lymph. It's going to try and bring it back into the bloodstream to the heart. Your lymph nodes are going to have lymphocytes in them. The lymphocytes lie in the lymph nodes to catch foreign antigen. Each lymph node will receive lymph from numerous vessels. So they're a filtering station for these vessels. Your red marrow is going to contain stem cells that will give rise to the cells of specific immunity. Your lymphocytes are the smallest of the leukocytes. They're going to include B and T lymphocytes. They're going to have large round nuclei with a thin rim of cytoplasm around the outside. They will act against specific pathogens, and they look identical under the microscope. So when you look at them, you can't see the difference between a B or a T cell under the microscope. B lymphocytes or B cells are going to mature in the red marrow of adults. They're identified by surface proteins that are found primarily in the spleen, lymph nodes, red marrow, and the Peyer's patches. Only a few of these are going to circulate and secrete antibody. Your plasma cells are going to be B cells that are actively fighting exogenous antigens. Your antibody or immunoglobulins are soluble proteinaceous antigen binding molecules. And the T cells or T lymphocytes, these begin in the red marrow and then travel to the thymus to mature. Your thymus is located in the chest just above the heart. It's most active in early life and begins to degenerate in adolescence. When we look at the lymph node structure, it consists of a medullary maze of passages that are going to filter the lymph and house leukocytes that will mount an immune response against the antigens. It will have a cortex, which is the outer portion. It's going to have a tough capsule that surrounds the germinal centers. Your germinal centers are where the cells of specific immunity are going to replicate. Other lymphatic tissue, we have mucosa-associated lymphatic tissue and other lymphoid tissues and organs, including the spleen, tonsils, appendix, Peyer's patches in the stomach, and lymphoid tissue in the respiratory tract. Antigens are going to be molecules that trigger a specific immune response. This actually means generates antibody. The epitopes are your antigenic determinants. It's the three-dimensional shape of the regions on the molecule that the body recognizes. When it comes to antigen size, larger molecules tend to make better antigens. Molecules that are too small to be detected can sometimes become antigenic when they're bound to larger carrier molecules, which are often proteins. Haptens are smaller molecules that are too small to be detected. They can become antigenic when they're bound to larger carrier molecules. Your exogenous antigens, these are going to be microbes outside the cells of the body. It will include toxins, secretions, components of cell walls, membranes, flagella, pili, and some DNA that gets released from the dead bacteria. Endogenous antigens are going to be microbes that reproduce inside the body cells. So they're hidden from the immune system, and the immune system can only respond if they're incorporated into a cytoplasmic membrane. Autoantigens are going to be antigenic molecules that are found on individuals' normal uninfected cells. These are normally eliminated during the development of the immune system and should be. Cell-mediated immunity is controlled by T cells. It's going to act against intracellular invaders, where antibody immune responses, or humoral immunity, is going to target extracellular invaders. So this came from the term humoral, which means body fluids. So these are going to be carried out and controlled by the B cells. So in preparing for an adaptive immune response, the MHC is important. This is your major histocompatibility complex that allows for recognition of things that are self-coded by a cluster of genes on chromosome 6. Self-tolerance means that you're going to have immune cells that will recognize autoantigens or self-antigens, and these are going to get eliminated during the development of the immune system so that you don't mount an immune response against self cells. There's two classes of the MHC here. The class 1 MHC is found on the cytoplasmic membranes of all human cells except the red blood cells. Class 2 MHC is found on antigen-presenting cells and B cells. Antigen-presenting cells regularly present antigen to the immune cells. These can include macrophages, dendritic cells, monocytes, and their relatives. And the cytoplasmic membranes of the antigen-presenting cells have about 100,000 class 2 MHC molecules. Epitopes that cannot bind an MHC typically don't trigger an immune response. So the MHC really is what determines which epitopes trigger the immune response. In clonal deletion, the body eliminates cells which are self-reactive since the receptors are randomly generated. In apoptosis, a lymphocyte is going to bind an autoantigen and it will respond by killing itself. So your HLA is the human lymphocyte antigen. These are glycoproteins that are found on the cell membranes of most cells. These were first identified on leukocytes in humans. Another term you may see is the CD, which stands for the cluster of differentiation. These are integral surface glycoproteins, and they were numbered in the order of discovery. So after we have an antigen, we're going to need to process it. So in antigen processing, this occurs before the MHC can display the epitopes. The antigen must be processed to isolate the epitopes. So when we process endogenous antigens, a few molecules of polypeptides produced by the intracellular invader are catabolized into smaller pieces that are about 8 to 12 amino acids long. And then they're moved into the endoplasmic reticulum. Here it's going to bind the grooves of class 1 molecules, and the membrane that is loaded with the class 1 gets packaged into vesicles that will fuse with the cytoplasmic membrane and displays the MHC1 protein epitope complex. So all nucleated cells are going to have MHC1 and can do this. When it comes to processing exogenous antigens, only antigen-presenting cells are going to be able to process exogenous antigen. The dendritic cells will phagocytize the pathogen and catabolize it to produce epitopes within a phagolysosome. A vesicle that contains class 2 MHC is going to fuse, and the MHC2 binds the complementary epitopes. The vesicle will then fuse with the cytoplasmic membrane and leave the MHC2 epitope complexes on the surface. And then the empty MHC2 will degrade. So your T cells are going to be lymphocytes that leave the marrow and mature in the thymus. A TCR is a T cell receptor. There's two different polypeptide chains with a groove that will act as an antigen binding site. It binds a complementary antigenic determinant. There are at least 10 to the 11th different sites. So your CMI or cell-mediated response, here the T cells are going to act directly against the antigen. They don't secrete antibody. This is used on intracellular invaders. So your TC is your cytotoxic T cell. It has hundreds of copies of unique TCRs and the presence of CD8. It will directly kill other cells. So those infected with the intracellular pathogens and abnormal cells can be eliminated. Your T helper cells are going to be CD4 positive. They'll have the CD4 marker. They help to regulate the activity of B cells and T cells. They can secrete various cytokines, which will determine which immune response will be activated. So Th1 is one subset of the T helper cells. These will assist the cytotoxic T cells in an infection response. They're CD6 positive, and they have a cytokine receptor called CCR5. The Th2 function with the B cells. They're more involved with hypersensitivity and allergy responses. These will have the cytokine receptors CCR3 and CCR4. During clonal deletion of the T cells, the body eliminates cells which are self-reactive. Since the receptors are randomly generated, you're going to end up with some of those. So a few self-recognizing T cells will remain alive to become TR or regulatory T cells. Those regulatory T cells, formerly called suppressor T cells, are both CD4 positive and CD25 positive. They're activated by other immune cells and will secrete different cytokines than the T helper cells. So when we look at the B cells, we have the BCR or B cell receptor that's going to be on the surface of the B lymphocyte, and it's covered with about 500,000 BCRs that are unique to that cell. It's an immunoglobulin made of 6 polypeptide chains. There's 2 heavy chains, 2 light chains, and those are going to form a Y. And then there are 2 additional chains that attach the Y with the antigen binding sites. It's estimated that each person forms at least 10 to the 9th and possibly as many as 10 to the 13th B lymphocytes, each that are going to have a unique BCR. So when the antigenic epitopes stimulate the BCR, the cell responds by forming plasma cells to secrete immunoglobulins. Antibody that remains integral to the cytoplasmic membrane will be produced. Each B lymphocyte has 250,000 to 500,000 identical copies. It has 2 antigen binding sites like other antibodies, which are randomly generated. So when you look at B cell activation and clonal selection, your antigen presenting cell is going to present an antigenic determinant to the T helper cells. This will activate the T helper cells with the complementary binding sites. The Th1 has a unique TCR and is CD4 positive. CD4 binds the MHC2. So in differentiating the Th cell, the antigen presenting cell signals the T helper cell with IL-1 to become differentiated to Th2. During clonal selection, only the B cells complementary to the Th2 are recognized, and they'll bind IL-4 to the activated B cell to proliferate and produce memory B cells and plasma cells. Your plasma cells are large activated B cells that are only going to produce the antibody identical to the parent cell's B cell receptor. It produces thousands of antibodies. The secondary cells are short-lived, usually only for a few days. The antibody can remain for months, and then the clones can persist for years. Memory B cells don't secrete antibody, but they will survive with the B cell receptors that are complementary to that specific antigen. They can live for years in the lymphoid tissues. The clonal deletion of B cells occurs in the bone marrow, and it's similar to the deletion of the T cells. Your self-reactive B cells are going to become inactive, or they're going to have to change their BCR. Antibodies are the free immunoglobulins. They're not attached to a membrane. They're going to have a similar to a BCR in their shape. So when we look at the antibody structure here, this is an example of immunoglobulin G1. The FAB region is going to be the arms of the antibody that are going to have the antibody antigen binding site. So here's where you would have those. The heavy chain are the 2 identical longer chains. They're called the heavy chain because of a heavier molecular weight. And the light chain are the 2 identical shorter chains that have a lighter molecular weight that are going to attach to the Y shape. Your FC region, this is where you're going to have the antibody stems. They form a fragment that is crystallizable. This is formed from the lower portion of the 2 heavy chains, and it gets named gamma, alpha, mu, epsilon, or delta, which ends up becoming IgG, IgA, IgM, IgE, or IgD. The variable region is going to be the arms of the heavy chain and the light chain that will vary in their amino acid sequence. They will vary B cell to B cell, but will be the same from any given B cell. Your antigen binding site is going to be the 2 variable regions that will form that antigen binding site. They're also known as the FAB regions, fragment antigen binding, and they're going to be complementary to the antigenic determinants or epitopes. So when we look at antibody function, one thing it can do is neutralization. It can bind to a critical portion of the toxin so that it can no longer function against the body. It can also block the attachment of molecules onto the surface of bacteria or viruses. It can also do opsonization. Here's when an antibody acts as a molecule to stimulate phagocytosis. So it can enhance phagocytosis. The neutrophils and the macrophages have receptors for the Fc region of IgG, and they'll phagocytize the antibody in the antigen it carries. So other things it can do is activate complement and stimulate inflammation. During agglutination, you'll have several immunoglobulin molecules that will bind with two antigenic determinants at once. This clumps the organism and the antigens together, and it hinders pathogenic activity and increases the chance of killing the phagocytes. With killing by oxidation, you can have catalytic properties, and that may kill bacteria directly. It can catalyze the production of hydrogen peroxide, ozone, and other potent oxidants. With ADCC, it's antibody-dependent cell-mediated cytotoxicity. This can coat target cells and bind epitopes all over the surface. The stem binds the receptors on the natural killer cells, and it will lyse the target with perforin and granzyme. These will be used to trigger apoptosis. It is similar to opsonization, but with opsonization, you're going to enhance phagocytosis, and with this, you're going to trigger apoptosis instead of phagocytosis. So it triggers apoptosis in this one instead of phagocytosis. When we look at the classes of antibodies or immunoglobulins, we've got 5 of them. IgG is the most common. It's about 85% of the serum antibody. It's involved with complement activation, agglutination, opsonization, neutralization. Its small size allows it to leave the blood vessels, and it will also bind pathogens before they enter the circulation. This one is actually small enough to cross the placenta. IgA, in many cases, is secretory. It's 5% of the total serum antibodies. Here, the monomers are connected with J chains to make another polypeptide in the various mucous membranes for a secretory component. So this would be found in places like tears, milk, saliva. It helps to protect the mucous membranes. IgM is the second most common. This is found during the initial stages of the immune response. It's 5 times larger than IgG because it's a pentamer that's held together with polypeptide J chains for joining. This makes it more efficient because of having multiple antigen binding sites. Your IgG is less than 1% of the serum antibody, so it's not important for agglutination, opsonization, or neutralization. But it does act as a signal molecule for the basophils and mast cells to trigger histamine release. This is going to help mediate between specific and non-specific responses, be involved with allergies and parasitic worms. IgD is not secreted by membrane-bound antigen receptors. It's often seen in the initial phase of the humoral response, but not in all animals. Its complete importance is not known, but it is involved with B-cell activation. So with class switching, this is the process where the plasma cell combines its variable region gene to a gene for a different stem, and it secretes a new class of antibody. The most common switch is to become IgG. So cytokines, these are soluble regulatory proteins that will act as intercellular signals when they're released by certain body cells. So the cytokine network is a complex web of signals between all the cells of the immune system. Your interleukins, or ILs, these are about 27 different types. They provide signals among the leukocytes. Interferons, these are antiviral proteins. They may also act as cytokines. And this would include something like gamma interferon. Growth factors, these are proteins that are going to stimulate cell division and ensure adequate leukocytes. TNF is your tumor necrosis factor. This gets secreted by T cells and macrophages. It's used to kill tumor cells. It also helps to regulate immune responses and inflammation. The chemokines, these are signals to leukocytes to go to the site of inflammation and activate other leukocytes. So a cytokine storm occurs when the feedback loop gets out of control. This will result in the harmful overproduction of cytokines. This appears to be at least a factor in certain diseases. So in cell-mediated immunity, here's where you're going to fight intracellular pathogens in abnormal cells. Your M cells or microfold cells are gateway cells that are located in the gastrointestinal tract. They're usually over the Peyer's patches, and they're adapted to be able to take up antigens from the intestinal tract. It will then transfer them to the lymphocytes and antigen presenting cells. So the activation of the cytotoxic T cell clones and their function, first you're going to have antigen presentation. Your infected dendritic cells are going to migrate to the lymph nodes and process the antigen and then present it with the MHC1. Next is helper T cell differentiation. Your CD4 positive T helper cells are going to bind to the MHC2 of the dendritic cell, and it presents the same epitopes on the MHC2. It causes the antigen presenting cell to more vigorously signal the cytotoxic T cell. During clonal expansion, it imparts a second signal in conjunction with IL-2 from a Th1 cell, and this triggers the division of the cytotoxic T cell to produce memory cytotoxic T cells and more cytotoxic T cell progeny. So then we end up with self-stimulation. This is going to be when you have daughter T cells that will be active and they no longer require an antigen presenting cell or a Th1. So this self-stimulation is an important part of amplifying the response. So the cell-mediated immune response is when the cells synthesize proteins. They display the fragments of the proteins in the antigen binding group of the MHC1, including viral proteins. Certain antigen presenting cells are going to secrete IL-12 to activate the T helper cells to become Th1. Th1 cells are going to secrete IL-2 and gamma interferon to activate the cytotoxic T cells with the TCR that's complementary to the antigen. These activated cytotoxic T cells produce memory T cells and more cytotoxic T cells that will produce IL-2 and the IL-2 receptors. So it becomes self-stimulating. It's no longer needing antigen-presenting cells or T helper cells. So the activated cytotoxic T cells bind infected cells with its TCR and with CD8 to its MHC1. It will then kill it through the perforin granzyme pathway or the CD95 pathway. So perforins are a cytotoxin that will aggregate into a tubular structure of an infected cell membrane. The granzymes are going to move through that perforin channel into a target cell, and they'll activate apoptosis. Then the cytotoxic T cell can move on to another infected cell in the perforin granzyme pathway. With the CD95 pathway, you have CD95 that's present on most cells. Cytotoxic T cells have a CD95L receptor. So when the CD95L binds CD95 on the target, the enzymes will be activated for apoptosis. Memory T cells, these are going to be made from activated T cells. They can persist for months to years in the lymphoid tissue. Subsequent exposure to the antigen matching the TCR responds immediately without an APC to produce the cytotoxic T cell, so it creates a much more efficient response. The regulation and co-stimulation is there to prevent the T cell from responding to autoantigens. It requires an additional signal from an APC or it will not respond. The TR or T regulatory T cells will also modulate your cytotoxic T cells. So in your antibody immune response or your humoral immunity, this is going to be the response against exogenous pathogens. So your T-dependent antigen is going to require a T helper cell for assistance. So a T-independent immunity is going to involve a large antigen with repeating antigenic determinants. B cells can bind directly to the large antigen, And the cross-linking of the repeating subunits to numerous BCR stimulates the proliferation to make the plasma cells and the antibody without the help of T cells. With T-dependent immunity, this is going to be most antigens. They're going to be smaller with less accessible antigenic determinants. The B cells targeted cannot function without the help of the T helper cells. So in looking at the inducement of the T-dependent antibody immunity, the antigen is presented for the T helper cell activation and cloning. It takes about a day for the T helper cells to survey the antigen presenting cells in the area for complementary epitopes with MHC2. The APC imparts the second signal to induce the T helper cell to proliferate by displaying the integral membrane protein. Next, we'll have the differentiation of the T helper cell into a Th2 cell that's signaled by IL-4. Next will be B cell activation, where the Th2 binds the B cell with the complementary MHC2 protein epitope, and clonal selection occurs. So here you'll have selected B cells that will proliferate as plasma cells to secrete antibody, and some will make memory B cells. Your primary response is going to generate relatively small amounts of antibody. It may take days or weeks to eliminate the toxoid or the antigen from the body. During the secondary immune response, your memory B cells are going to make a reserve of antigen-sensitive cells that will activate with another exposure to that toxin or antigen. And then the cells proliferate and differentiate rapidly without the interaction of the antigen-presenting cells. So this will result in large amounts of antibody within a few days. It's faster and a more effective response. The memory responses are going to be your enhanced immune responses to the subsequent exposure to the antigen. Acquired immunity is going to be immunity that develops with exposure. So naturally acquired active immunity, the body responds to the antigen and pathogen with a specific immune response. So it would be exposure from the environment. Naturally acquired passive immunity, this is going to use IgG that crossed the placenta and IgA from the breast milk. This will provide early protection to infants. It's passive because the child is not actively producing its own antibody. With artificially acquired active immunity, the immunity is induced by introducing an antigen in the form of a vaccine. Here, the patient's own immune system will actively respond. With artificially acquired passive immunity, this is used when the active immune response is too slow, something like snake venom, where you're going to need to have an immune response happen very quickly, faster than the body will do naturally against the toxin. So antibodies are harvested from the blood of immune humans or animals, and then it gets injected as an antitoxin or an antisera into the person who needs it. So this is the end of this section.